thank you for joining us today. I am very pleased and happy to welcome today Dr. Michèle Desjardins. Driven by her bachelor's degree in physics, Dr. Desjardins obtained a doctorate in biomedical engineering and joint supervision from Université Pierre et Curie Paris 6 and l'École Polytechnique de Montréal. She then went on to complete a postdoctoral fellowship at the University of California, San Diego. She is now a assistant professor in the Department of Physics, Engineering Physics, and Optics at Université Laval, and a researcher at the CHU de Québec-Université Laval Research Center. Her research focuses on in vivo imaging of blood vessels and oxygenation. And today, she will tell us about the multiscale neurovascular imaging and modeling in the mouse brain. So please welcome me and join me today in welcoming Dr. Michèle Desjardins.
Hi, thank you for the introduction. Thank you for the invitation to give this talk. And yeah, I'm going to be telling you about my research. I think I made this talk kind of oriented towards students, which I think was a good bet because it seems like there's the majority of the audience, meaning that I'm explaining maybe a little bit more about the tools. That's also because I'm a physicist, so I tend to be excited about the tools and not only the biological questions. And I'm going to tell you a little bit about my story because, well, those that I've met during lunch, you know that I like to give advice to thinking about my past self as a student. So let's just do that. So starting back in 2007, when I was a young physicist at this point, I always knew that I wanted to do a PhD because both my parents were professors, but I just didn't know in what. So I had studied physics and I liked my math and my equations and my programming, but all the different fields of physics that I tried during my undergrad, I wasn't like passionate enough about to pursue. And in the end, it's my mom. She's a psychoanalyst and she was reading this book on neuroscience and psychoanalysis. And that introduced me to the entire field of neuroscience, which I decided to pursue here in Montreal, actually, at Polytechnique. And so when I met my advisor, he proposed a project using rats, like studying the rat brain. And I was like, I don't want to study rats. They're all going to be thinking like cheese, scary cat, and boring things. I wanted to study complex human brains with like emotions and memories and the unconscious. I was all about the psychoanalysis theory. So I refused the rat project and I decided to study human brains. So I discovered this super cool technology that we're using to study human brains, functional MRI, which is basically where, as you all know, stick someone in a scanner, some sort of magic happens, and you get images of the brain without even touching them, especially images of brain activity. And now, what I also discovered, I don't think I have to explain to you guys, but is that what we're actually imaging when we call it brain activity is that the level of oxygenation of hemoglobin in the blood. That's what we're really seeing with MRI because that's what interacts with because the paramagnetic deoxyhemoglobin disrupts the magnetic field, makes it less homogeneous, and that changes the signal that we measure with functional MRI. And then why is hemoglobin related to brain activity? Because of neurovascular coupling. So because when neurons activate, they consume oxygen, they get thirsty, so they scream at the blood vessels with some molecules that they need extra fresh blood to bring them more oxygen. And so that's what we call neurovascular coupling. And so in summary, what I found when I started in this field was that our magical, super great tool to study brain activity actually only sees it through a filter of neurovascular interactions and at a different, both spatial and temporal scale, because neural activity per se happens on a micrometer and microsecond scale, millisecond scale. And then what we're measuring it with fMRI is on a scale of millimeters and seconds. So this led me on a quest to better understand those neurovascular interactions and across different spatial scales. And so the rodents that I had wanted to leave behind, they ran after me and they just came back in my life. And I've been stuck with the mice ever since. So I turned to mice to study such interactions because of all the tools that we have in mice that we don't have available in humans. And now I'm going to tell you about those tools that we have for measuring neurovascular interactions across scales. So in mice, we can manipulate neural activity using optogenetics. I'm not going to say a lot about this. And we can then measure microscopic scale, so single cell, single vessel activations using two-photon microscopy. So that's a very common technique, but I'm considering that I'm speaking to an MRI community, I'm going to still introduce it. So in mice with cranial windows, we have to express a contrast agent, which can be genetically encoded or injected. And then by mechanically scanning a laser focus point in 3D, we get a 3D image with micrometer resolution depending on your contrast agent, could be of neurons or vasculature. So here, for example, we have the vasculature in like maybe half a cubic millimeter of tissue from the surface, larger vessels on the surface diving down and the capillaries in between. Now, if we look at just one plane of this 3D image, for example, if we're imaging neurons, the most popular agent for that is the calcium indicator GCAMP. So basically, the more the neurons fire, the higher the calcium concentration and the higher the fluorescence of this contrast agent. So it can be expressed specifically in different types of neurons. Here it's just expressed in all the neurons, and I can zoom in individual neurons and look at the activity in each neuron. For example, here, every time we stimulate the whiskers of the mice, there's an increase in the fluorescence that we measured that was in the barrel cortex. Then if we inject the contrast agent in the vasculature, we get images of blood vessels looking something like this, and we can zoom in on one individual vessels and see the individual red blood cells. So we could have a measure of the red blood cell flux or individual vessel blood flow. And finally, there's a contrast agent or a family of contrast agents that are sensitive to oxygenation. So we can have measures of absolute partial pressure of oxygen in either the vasculature if you inject it in vasculature or in the tissue surrounding. So our data with two-photon microscopy can look something like this in a combined experiment where we have a video of the activity of neurons in green and blood vessels in red. And every time there's a flash and increase in fluorescence, it means there's a burst of activity. Or we can also take structural images of the vasculature, which we call angiograms with two photons. So here it's the video as we're starting from the surface and going deeper down towards the depth of the brain. And we're seeing each blood vessel, so surface blood vessels, and here are the capillaries. The field of view is about 600 microns here. So that's what we can get, very detailed measures that we can get at the microscopic scale, but still that's less than the cubic millimeter of tissue. So even for a mouse, that's very small. So we also turn to a mesoscopic scale imaging technique, which is wide field imaging. Again, we combine different contrasts to have both neuronal and vascular oxygen contrasts. The main principle of wide field imaging is really simple. You just have to film. So you don't even need a cranial window for this. You have to remove the skin, but you could image through the cranium. You just really film with a camera, and depending on which light you illuminate with, you'll get a different contrast. So in the same way that oxy and deoxyhemoglobin have different magnetic properties, they also have different optical properties. So we use that to reconstruct their relative concentrations. They're just relative changes in oxygenation. Then using scattering as a contrast. So in two-photon, scattering is your enemy. It prevents you from imaging deeper than like 0.5 millimeter or maximum 1 millimeter in the cortex. But in laser spectral contrast imaging, it's your friend because it's actually the scattering by red blood cells makes the image more blurry. And so we quantify this blurriness, and where there's more flow, there's more blurriness. So we get blood flow contrast. And finally, the same calcium indicator that we use in two-photon can be used in wide field imaging to get neural activity. And this could be cell-specific. So we could express the calcium indicator only in excitatory or inhibitory cells or any sort of genetic combination that we can imagine. So our data sets look something like this. This is through a cranial window. of about like 7 by 7 millimeters. So I would say we get about half the cortex, the cortical surface with this, which what I call mesoscale imaging. So we have some of the barrel cortex, motor cortex, a little bit of visual, a little bit of retrosplenial, which is like I'm not sure exactly, another part of cortex. So we don't have the whole cortex, but we have a good amount, good enough to study network scale questions. And we have a contrast where we have simultaneously blood flow, neural activity, blood volume, and HBR, which is what we call our proxy for measuring bold. So it's the closest we can get without bringing a mouse in the fMRI scanner to measuring fMRI in the cortex. And this has a temporal resolution of, well, about 10 hertz, but all the contrasts here have timescales of being vascular contrast, or the calcium activity also have timescales of seconds. So we actually have too much of temporal resolution for the intrinsic timescale of what we're measuring. We also monitor the mouse during this imaging. That's easier than in a scanner, because we just have to put any kind of camera in front of the face of the mouse, and we can look at the mouse level of arousal with the pupil diameter, face motion, and eventually there's some tasks that we can have the mouse do and monitor its result. But obviously the ultimate translation would be to measure the same thing in mouse as we measure in humans. So we would like to bring the mouse inside the MRI scanner, but for that we had to ask the mouse to kind of stay really still so we don't get like motion artifact in fMRI. And which they kind of did thanks to the help of this kind of contraption where we made this to head fix the mouse inside the scanner. So that was this one we made during my postdoc. Believe it or not, it looks better than the other one we did. So that's why I'm showing this one in my talk. It's just really, so this is the mouse bed that we used to bring the mouse in the scanner, where we have head fixation. We brought a little tube for air puff stimulation. We had an optical fiber. We were using it for optogenetic stimulation, but other groups have used it for optical imaging. So you could use it to bring light, but also to collect light. We had a reward, so we could have some sort of behavior with the mouse, just bringing a tube to the mouse's mouth, and a monitoring camera. And we had to make our own coil because this whole thing wouldn't fit in the regular coil. So with this kind of, combining all these techniques within one mouse, we can study questions where we're interested, for example, in cell type specific neurovascular coupling, and then we look at individual vascular responses in single arteries or single veins. Then you can look at the cortical surface of your contrast, where you can separate neuronal and vascular contrast, and then you bring the same mouse in the MRI scanner and look at what you would measure in a human under the same conditions. So that's like a framework, basically, that we are developing. Other groups have combined, like I said, the wide field imaging simultaneously with the scanner. So they'll bring a bundle of optical fibers where each fiber basically is like a pixel of our camera inside the scanner, and do simultaneously the MRI and the optical imaging. That would allow you to study event specific, like for example, resting state neurovascular coupling, where you really have to measure simultaneously the neuronal and the vascular sites. And one thing that we did with this framework was studying the cell type, the cell specific signatures in the bold signal. So we had 2 mice lines with which we were doing optogenetics. So in one mouse line, excitatory neurons were expressing channel rhodopsin, meaning that by shining blue light on their head, we would excite specifically excitatory neurons, and in the other line, inhibitory neurons. So we were looking at the time course of the bold signal, not the spatial, no spatial information for now. So what we found was that for both shorter and longer stimulus, if you activate only excitatory neurons, you'll get some sort of initial dip plus an increase and going back to baseline directly, while inhibitory neurons also have an increase, so they don't lead to negative bold, they lead to a positive bold, but then followed by an undershoot going back to baseline. So I know that the question of the undershoot, that origin of the undershoot was a big question when I started my PhD, but now I kind of left the MRI community, so I don't know if people even talk about this anymore. But back then it was kind of cool. So there's that. And other groups are studying the more like molecular or the subtypes of cells that are responsible for specifically this undershoot or this constriction, vascular constriction following the initial increase. That's not my, I'm no biologist. But that could be useful. This kind of info could be useful for getting more information on excitation and inhibition just from measuring BOLD. So, for example, the excitation inhibition balance in different regions may be reflected in the shape of the hemodynamic response. Now, all the methods that I've mentioned up until now have different limitations, obviously. So in 2-photon, like I mentioned, we're scanning a laser point by point in the tissue and collecting our 3D image point by point. So a 2D plane is pretty fast. It's like 1 second, but then for going in depth, you have to physically move the sample. So that takes a long time, and a whole 3D image takes minutes. So you cannot do functional imaging in your whole field of view with 2-photon. You'll always have a trade-off between the field of view and the temporal and spatial resolution. With the wide field mesoscopic imaging, we have no depth resolution. We're only seeing the surface of the cortex. And with fMRI, obviously, you don't have any neuronal information and you don't have single cell resolution. So we're left with this gap between the scales that we're trying to fill in my lab, in my group, to study neurovascular interactions across scales and to fill the gap between the scales. We're doing this both with some experimental and some modeling things that we develop. So I'm going to start with some experimental development. How can we improve the limitations of 2-photon microscopy? So classic 2-photon microscopy uses a normal laser focus. That's just a point. It's a Gaussian focus. So it means that you scan in 2D, then you move the sample, you scan again in 2D, and you get a collection of 2D images that constitute your 3D image. If you, instead of using a normal Gaussian beam, you spread out your beam like a line, then you can scan only in 2D and get the signal from a 3D region in the brain. So this means you're much faster. You're getting your whole 3D volume, you're imaging your whole 3D volume in the time of just doing one plane from the normal focus. So you're losing your depth information, but if you can combine both the Gaussian and the Bessel beam in the same microscope, then you can compare the two images to tag each vessel with its depth information. And then you can get about like 50 to 100 times increase in scanning speed, and eventually you can get some functional imaging with 2-photon, with cellular resolution. Another technique that we're developing is OCT, optical coherence tomography. The principle is really like an ultrasound, but with light. So you're sending a pulse of light, and then every time there's an interface, a change of refractive index, you'll have an echo. A part of the light is reflected, and then you can measure it, and the timing of the echo tells you about the depth of each interface. Because it's light, it's so fast, we have to detect it with interference, but the rest of it is the same as ultrasound. So with one measurement point, you get a depth profile, and you only have to scan in 2D to get a 3D image. And so these are not my images because we're still building the system in my lab, but depending on your imaging parameters, you could have a contrast where you see neuronal cell bodies, or if you take two consecutive images and subtract them, so you'll have only, you'll remove the signal from the static tissue, you'll only get signal from the vessels, kind of like in ASL, arterial spin labeling, kind of the same principle. Then you get a contrast where you see the blood vessels. And with Doppler OCT, you can get images of flow throughout the field of view. So that's what we're trying to build in our lab. That's actually an actual depiction of our lab that my students made. I think they have too much free time. So now that was about the optical imaging techniques that we use in our lab. Now I'm going to tell you about the modeling techniques that we use for analyzing our data. So I'm going to tell you about 3 specific projects where we use data from all the different scales. Some of them are actually, many of them, especially the mesoscale, are ongoing. So I'm just going to tell you about our hypotheses, and then you'll have to come back in 2 years to see the results. So as far as the microscopic scale goes, the point here is to use our knowledge of MR physics to construct a model so our knowledge of MR physics and biophysics really, to simulate MR signals, functional MRI signals from vascular structure. So we measure with 2-photon, we measure microscopic images of blood vessels. And then we know that in these blood vessels, there's deoxyhemoglobin that is what is causing our fMRI signal. So we're going to try and model all of this. So the first step is to turn this image into a vascular graph, which really is just a way. So graphs are tools from the network science theory, which in this particular case, we're just using the graph as an object to package the information that we have in our image. So there's no graph theory implied. It's really just a packaging of information. So we use machine learning to automatically segment the images and then skeletonize them and label each vessel. So we now have the information of each vessel. Where is its position? What's its diameter? And what does it connect to? So each branching point and what other vessels does it connect to? We have all this package in our graph, from which we can extract some features like the radius, length, number of connections of each vessel, which can come in handy for biological questions, for example, if we want to quantify vascular differences between different mice. So from this vascular graph, we're just going to apply some basic plumbing rules. So you have a pipe split that splits into two. Half of the flow goes in each arm. But if one of the arms is twice smaller, then you have 16 times less flow going in there. That's Poiseuille's law. So we applied this at every branching point in our graph to get a map of blood flow. It's a map relative to the input. So you have to have like an initial condition to get an absolute flow map. And then from here, we model oxygen diffusion in the vascular tree. So oxygen is tied to hemoglobin and is transported through flow. And then it's in equilibrium with oxygen that's just freely diffusing, dissolved in the blood, and diffuses through the membrane to go to the tissue. And we have to solve this iteratively because of this equilibrium between the two. And so we use finite element methods to make it faster in the tissue. And then so from our map of blood flow, we get a map of oxygen. In itself, that's very useful. So we could stop our model right here because there's currently no method that allows us to measure oxygenation, absolute oxygenation with this level of resolution throughout the field of view. So I mentioned that we have an oxygen contrast in 2-photon microscopy, but you have to measure point by point. So you can only get like maybe 100 different locations, big maximum in a single experiment, and that would take like 2 hours. So being able to get a full field of view, in a field of view as big as our vascular image is, a map of oxygen is already useful physiological information. And so one thing we're doing with this is trying to use it to model oxygen in tumors. So we've been implanting tumors in mice brains and trying to image the vasculature. It doesn't work with 2-photon because the vasculature in tumors are too leaky. So the contrast agent is not going to stay. But with OCT, we're hoping we're going to be able to image it. And the goal of this is that in tumors, when you treat a tumor with radiotherapy, the regions of the tumor that are hypoxic, that don't have enough oxygen, they resist the treatment. So they don't, like radiotherapy doesn't work on them because you need the oxygen to fix the damage by the radiation. So we're thinking that if we can optimize the dose distribution of the radiotherapy towards the region of the tumors that are oxygenated, then we're going to maximize the efficiency of the treatment, radiotherapy treatment. So that's where we think that we're trying to make a proof of concept in a mouse that if we have vascular images and we can model the oxygen, we can optimize radiotherapy treatment, which obviously eventually could have clinical applications. But then again, why were we modeling oxygen in the first place? Because that's what actually we're measuring when we're measuring functional MRI. So from our oxygen map, basically, which is a HBr map, deoxyhemoglobin, we model how it disrupts the magnetic field because of its paramagnetic properties. And then we using Monte Carlo, we model a random walk of protons in this inhomogeneous magnetic field. And then we sum all the vector sum all the protons to model what we would macroscopically measure with MRI. And we get quantitative model of the MR signal as a function of sequence parameters. So when do we measure as a function of T, TR, and also as a function of physiological parameters that we can vary. So we've been using this framework for modeling specific MR sequences, for example, vaso, which is sensitive to blood volume. Basically, our paper was showing which combination of parameters you should choose to maximize your volume contrast and minimize your bold contamination in vaso. We also used it to validate some simplified bold models. So say you want to study the effect of your parameters on your signal, but you don't have time to do the whole Monte Carlo thing. Can you use the simpler bold models that have been like more heuristic models with just a couple parameters that have been proposed in literature? Or which parameter, or in another way, framed in another way, which details of our super detailed model are more important? Do we need this 3D geometry, or can we just model one artery, one artery compartment, one venous compartment? Do we need this detailed oxygen map, or we can approximate that the oxygenation is homogeneous? So we did this study. If you want to know about the results, go read our most recent paper in NMR Biomedicine. Yes, that's it. Here by Jordan Charret. So that was the microscopic modeling that we do. Well, microscopic to macroscopic, really. Now I'm going to talk about how we use the mesoscopic functional, the mesoscopic data for some biological questions. So we're going to start with a simple observation that the vascular network changes across lifespan. So, for example, these are images, 2 images of the vasculature in a 500 by 500 micron field of view in a normal adult mouse, where you see, so you have all the capillaries going in all the directions, and at the surface you have the larger vessels just parallel to the surface and then diving deeper down and separating into smaller capillaries. And then this is an image taken with the same technique in the same brain region, but in a mouse that's 2 days old. You see that it's very different. So all the vessels look like they have similar diameter. They also look, they're all parallel. So this is an image from the surface. So it's like they're all diving down and there's not a lot of connections between them yet. So there's obviously a lot of development of the vascular network that's ongoing at this age. And then during aging also, that's the same adult mouse at like 12 months old and then 18 months old. And there's obviously a lot of remodeling again of that's the surface vasculature. We see vessels become more tortuous, possibly a change in vessel density. So there's definitely something happening with the vascular network across lifespan. And not only the vascular network, but like many changes are accompanying normal aging, and there's a lot of literature about this. During aging, we see changes in functional imaging signals in their amplitude, as well as in the blood-brain barrier. The vascular network will become more tortuous, decrease in density, decrease in the reactivity. So like if you use a vasodilating stimulus, vessels will dilate less or more. It actually probably changes in a U shape during aging. Blood flow decreases with aging. That's been known in humans. That's all data from rodent studies, but there's also a lot of evidence from humans that blood flow is decreasing. Functional connectivity varies in a U shape. The network properties of networks vary with aging. And yeah, there's been evidence of hyperexcitability. And obviously there's changes in behavior and cognition during aging. So there's already a lot of literature, but most of it comes from studies at 2 time points. There's like a young group, an old group. If you're lucky, there's a middle-aged group, but really not that many people have used longitudinal imaging, maybe because the optical techniques that I've mentioned, it's not been that long that they've been available with the chronic cranial window preparation that allows to do longitudinal imaging. It's been maybe almost 10 years, and still we haven't seen that many longitudinal studies. So we're starting such a study, like I've told some of you already. with our hypothesis being that we're going to observe the mice from 6 months old until they die, which should be around, hopefully, 24 months. So we're thinking that initially neuronal hyperexcitability will lead to an increase in oxygen demand. So that would decrease oxygenation. And this is all things that we can measure with our 2-photon measures. But that initially there's going to be compensatory mechanisms where vasculature is going to compensate for that. And so we're not going to see changes yet in the behavior. And then at the mesoscale, we can also look, while looking at neural activity and microvascular properties, we can also look at the resting state networks, such as we can measure them with our mesoscale imaging. So it's only going to be cortical resting state networks, which we can identify with the seed-based approach or any ICA or any type of approach that is used in MRI would also work for this data. And so eventually when plasticity in the vasculature starts to decay, then we're going to see changes in vascular reactivity, changes in the networks, in capillary density, which leads to hypoxia and the behavioral deficits. That's our hypothesis on what we're going to observe. So let's talk again in 2 years when these data have all been these mice have all been imaged and they rest in peace, and we can tell you if we were right or wrong. So that's just preliminary data showing that the older mice tend to stick to the walls more. If you just let them run free, they're going to tend to stick to the walls more than the younger ones, and that's associated with anxious behaviors where they're like more stressed. And also that in response to vascular stimulus, so it's a hypercapnia. It's also used in MRI, so you guys may know about it. So it's just supposed to not change neural activity, but just dilate the vessels. And it seems that the middle-aged mouse has higher dilation for the same stimulus compared to the younger mouse, but that's just n equals 2, so I would bet on it yet. I said there's going to be a lot of ongoing projects. So the second ongoing project that we're doing is with the mesoscale imaging, is studying the effect of neuromodulation. So that project started because I was at a conference and the flight got canceled coming back. So I was stuck at a cheap motel by the side of the airport with a colleague that I've never met. So we were like drinking this disgusting coffee. And, oh, hi. Oh, you're also at Laval. Oh, I don't know you. What do you work on? Oh, neuromodulation. Oh, I work on neurovascular coupling. Has anyone ever studied the effect of neuromodulation on neurovascular coupling? I don't think so. Sure, let's get a grant and do it. And we actually got a grant. So we're doing it. So that's really the principle. We built a system where we're going to simultaneously measure the activity in the locus coeruleus, which is where noradrenaline is released. And it's related to the arousal level of the mouse. We're also filming the mouse because this is like the pupil diameter is going to be a non-invasive proxy of arousal level, and then going to look at neurovascular activity. Again, we're still collecting data, but what we have been doing so far is using long-term short memory RNN model to see if we can predict the locus coeruleus activity from just the non-invasive measures from filming the mouse, so that eventually we can get rid of this fiber in the LC. And if we're doing this in humans, we don't need to implant them with fiber. We can just film their pupil. And it seems like in the mouse, the face motion predicts arousal level more, better than the pupil, but that's not going to work in humans. And so we do get a pretty good prediction, maybe like 50% of variance predicted in the LC activity. And it's been in I just wanted to name this preprint that I've seen in the same vein, where basically some people in this work, they are claiming, and it's not yet been published, it's just a preprint. I don't know the researchers, but they're claiming that just from the pupil diameter, they're predicting the entire, they're having a very good prediction of the entire neurovascular activity in the brain. So if you're reading any literature, you know that arousal and neuromodulation are buzzwords currently in the neuroscience community, at least. So there's that. So I'm just wondering at this point when I see this paper is like, do we even need to image the brain, or are we just gonna look at the pupil diameter and stop doing neuroscience? So that was fun, but I'm doing it all. I'm also getting on the train of the neuromodulation question. And we're another thing we're doing with this kind of mesoscale data is we're trying to model, so based on structural connectivity from atlases, we're trying to model the effect of neuromodulation on functional connectivity and match it to experimentally measured functional connectivity. That's also ongoing. I promise the next project is going to be an actual published project where it's not ongoing and I have answers, not only questions. And it's the one about macroscopic functional connectivity having a microvascular origin. So this is my a third coincidence that again steered my research towards something else. I have a colleague who was hired the same year as me. We both like good food and good wine, so we hung out, and our babies were born on the same day. Like, what are the chances? So he's a network scientist, not neuroscientist, just network science. Like, for the sake, he does super abstract network things like embedding them in hyperbolic spaces and things that I didn't know existed, but also some network neuroscience collaborations with me. So yes, here we're going to be interested about the link between structural and functional connectivity. So I think you're probably aware, but just to be clear, structural connectivity means physical structural connections between regions, such as what we can measure with diffusion MRI, and functional connectivity is what we infer from functional imaging. When two regions are activated together, we consider them functionally connected. And we can represent these in matrices, the functional connectivity matrix, for example. Here, if we have three regions, so all the rows and all the columns are all the regions, and then regions one and 2 are strongly connected, and then 2 and 3 are more weakly connected. So there's a smaller value in the matrix, just to make sure we know what we're looking at for the next slides. So there's been quite some interest in the literature in modeling the functional connectivity as a function of parameters from structural connectivity. There's a variety of papers doing this. This is from the group of Bratislav Misic. He's at McGill, so you probably know him. So they found that by considering some properties of the structural connectivity matrix, I'm going to explain communicability later, as well as the physical distance between regions, they could predict a significant amount of variance in functional connectivity. So meaning that if two regions are close by together, or if they're close by in the network space, really, like if they communicate easily in terms of the number of nodes between them in the network, then they're more likely to be activated together, so functionally connected. And they also looked at the spatial distribution in the brain of this relation, and they found that it varies according to the gradients of hierarchical gradients in brain activity from like a unimodal to more transmodal regions. If you know about it, then you know, and if not, if you know, you know, like they say. So in that vein, we started wondering if we could explain, and other people have also included other predictors instead of just structural connectivity, regions that are similar in terms of gene expression, for example, also tend to be more functionally connected. Some people call this homophily, so regions that are similar tend to be more connected. So we wondered, since this fMRI matrix, right here, it's measured using a vascular contrast, right? Bold fMRI actually is a vascular contrast. Can vascular structure explain some of the variance in functional connectivity? And we have, based on literature, we have evidence to think that, yes, vasculature does influence resting state activity and functional connectivity. The ratio of flow to oxygen metabolism determines the bold SNR. Cerebrovascular reactivity determines the SNR and the timing. And all these parameters will influence your measure of functional connectivity. Regions with higher flow have stronger functional connectivity. So we have strong evidence that there is an influence of vasculature on FC. So we set to use publicly available data to construct a linear model of FC to include structural, but also vascular properties, and see if we can increase the prediction of FC. This we can't do in humans because we don't have very detailed vascular maps in the atlases that we have in humans. We don't have any vascular information, or at least not microvascular information. But in mice, we do. We haven't for a long time, but since 2020, two groups have published these whole brain vascular atlases in the mouse. So they stained, it's acquired ex vivo. They stained every single vessel in the mouse brain, including down to the capillary level, and they labeled them into a graph. So we have these, and they're co-registered to the atlases. So we have detailed microvascular information from the mice that we could use. And these are so we did our whole model in mice. So for functional connectivity, we used two publicly available data sets of fMRI, and then we pre-processed it using rabies. This great toolbox. And then and we extracted the functional connectivity matrix as one usually does. Then we use the Allen Atlas structural connectivity data to extract a measure we called communicability. So that's really a measure of how much information you can pass from node A to node B. So for example, in this drawing, you have 3 paths from A to B. But so basically you sum all of them, but you penalize the longer paths by normalizing them to the number of segments you have to go through. So that's how we get a matrix of communicability. Then we defined vascular similarity, telling us how similar in terms of microvascular structure two regions are. And so we have a matrix of a value for how similar two regions are for each pair of region, how similar they are in terms of vasculature. And then physically, the Euclidean distance and spatial adjacency of two regions are also encoded in the matrix. So really just, this model is just telling us if two regions are close by together, if they're adjacent, if they're similar vascularly, and if they can communicate easily on the network, they're more likely to be functionally connected. And then we estimate the betas of the parameters using ordinary least squares. And the higher the beta, the more important the predictor is in predicting functional connectivity. So how did we design this vascular similarity? We defined a certain set of, so we extracted from the whole brain graph, we extracted the subgraph for each brain region, and we defined a certain number of vascular features. And then the distance in vascular feature space between the regions, the inverse of this distance is the vascular similarity. And what features did we use? We defined a whole bunch of them, and we picked the ones that were explaining the most variance, which were the capillary length, radius, average tortuosity, and branching point density. So it's really about microvasculature or capillary features that we're using. We constructed this model, like I said, using physical, structural, and vascular properties. And we explained, so this model in its whole explained 52% of variance in functional connectivity, comparable to other models that instead of vasculature have gene expression or other biological properties. And if we do the model just with vascular similarity, we explain 15% of variance in FC. So that's not negligible. There really 15% of variance between regions in FC is explained by just the fact that they're vascularly similar. Also, like this comes from 3 different data sets, right? So it's this FC data, the SC data, and the vascular data are all different data sets. So we're actually surprised that it worked so well. Then little anecdote about anesthesia. I think everybody knows that anesthesia has effects on the brain. So for example, this is just anecdotic data from our lab where we were looking at the structure of like the propagation of activity, of vascular activity after the mouse spontaneously moves its face. And in awake or very lightly anesthetized, you have this like propagation of a wave-like propagation of activity. And then under deep anesthesia, it just goes away completely. The structure goes away. And indeed, literature tells us that anesthesia has impacts on functional connectivity. and that different anesthetics have different effects. And I just heard that your group has recently is about to publish a paper about these effects. I'm very looking forward to seeing it. So we wondered how this the microvascular contribution to FC is going to change using anesthesia. And indeed, so the entire functional connectivity matrix is a little different. So for this, we redid the same analysis, but we used a publicly available data set acquired in awake mice. And it looks like the entire FC matrix has lower values in awake, which could be, anytime I compare awake and anesthetized data, I always say like, it could be because everything is less, under anesthesia, everything is more coordinated. There's less complexity to the activity, so everything seems more connected, but could also be, and it could be a little bit of both, that in awake data, there's a little bit more movement, so your SNR is less good, so you find a little bit less correlations. And I always say that when you're analyzing awake, it could always be a contribution. The motion artifacts, you would never get rid of them completely in awake MRI. But it seems that anesthesia increased the variance explained by vascular similarity and reduced that explained by Euclidean distance a little bit. So it's not super easy to interpret, because also like the anesthesia was a mix of two anesthetics, one who's a vasoconstrictor and one is a vasodilator. So it wouldn't be super straightforward to interpret. So this is available as a preprint and should be published anytime soon. But it's also an ongoing project where we're wondering, okay, microvascular structure explains some variance in functional connectivity, but is it just because FC is measured with fMRI, which is a vascular measure, or is there some biology under that? Is it because, for example, regions that are similar vascularly are more similar in terms of cell types, or anything else that could explain why they're more functionally connected? And to answer this should be easy, because we're able with our mesoscopic system to measure just neuronal activities without the vascular lens. So then we could look if the functional connectivity from just a neural activity is still correlated to microvascular structure, or if it isn't. That would allow us to distinguish between these two hypotheses. How does it vary spatially in the brain or under different states? Are there other? So we just look at FC, but are there any other properties of the signal, dynamic properties that can be correlated with vasculature? And then my big open question is, I've been using, in this work, we're using vascular networks, like vascular graphs, like I was saying, just as a tool for packaging information. But graph theory, like network theory, is a whole science where information is propagated in a network. And when you think about neurons, yes, they communicate information, it makes sense. But in a way, the blood is also information for the brain, for the vessels. So can we apply some of the tools of network theory to interpret blood as a signal that carries information and use those network tools to study vascular networks? That's a big, broad questions that I've been thinking about for some time. And how, if so, how, like to what extent can we do that? Anyway, we can chat about this. And thank you. I'm going to thank my collaborators, and thank you for listening. I wish you all a lot of fun doing some fun science.
Hi, thank you so much for showing us all these different projects that you have. At some point, you said something that caught my attention. You said that the cells show like they speak to the to the vessels. And I was wondering, when you were showing your aging projects, if rather than we have changes in the vasculature as you show, and is it possible, or do you know if also this calling or this communication with aging changes in terms of the intensity of the calling? Like, for example, the molecules that actually, like, I was thinking this because maybe
this signal is also diminishing as we age, and that's why we have this hypoxia or elimination of the vasculature. So I don't know, just a question.
So I don't think there's an answer yet to that. Like I don't think it's been studied specifically, but one thing that people see is that vessels are less good, or depending at which age, less or more good at dilating for an equal stimulus. So it could be because the molecular messengers are changing in concentration. I mean, it's still like what are the specific molecular mechanisms for neurovascular coupling is still an open question, even in healthy normal mice. So some people are still studying this question currently. So I don't think anyone has looked at it in aging, but it could be. And then there's definitely a change in the vascular response, but is it just mechanical because cells are less elastic or like less, or is it just because there's change in neural activity? I mean, it's probably many, as in everything, there's probably many contributions, but that's just an open question. And I'm actually not going to look at molecular messengers in this current study, but like I was saying, hopefully it gives us enough data to pursue this question with a lot of future funding. So maybe in a future project.
Hey, really good talk. Towards the end, you mentioned that the structural connectivity and I guess vascular structure explains over half the variance in functional connectivity in your mice. I was wondering about, I guess, the remaining 48%. Isn't that biologically what we care about? Where a lot of times, especially in the human literature, functional connectivity is incorrectly interpreted as being like, oh, like coactivity between brain regions and like some measure of like, I guess, neuronal coactivation. But I guess what you actually were able to do is disentangle the effects of that from just the vasculature. Like, would you think that using whatever's left over after regressing away vasculature and structural connectivity, do you think that remaining connectome would be useful for scientific questions, or is it just noise?
Oh, excellent perspective. I hadn't like thought about it this way actually. So just to clarify, this is a variance between regions. It's just we averaged over the mice the matrices, but that's actually a very good question. It's like we did this model. It's like when you analyze your fMRI data and you put nuisance regressors and you regress out the signal from movement and stuff, and then we could say that the vascular structure, we want to remove this, and then we just want to look at the rest. I think it could be interesting. I had never thought about it yet. Thanks for the idea. I think we can think about this.
Thanks for the great talk. You showed at one point the list of citations regarding anesthesia affecting the functional connectivity. Yeah, perfect, right there. Have you done similar kinds of work using either your 2-photon or your window-based imaging to see how those effects change?
I haven't at all. Like the only data that I have comparing anesthesia versus no anesthesia would be only with isoflurane first. Like that's all that I use really. And we just haven't looked. Like we use anesthesia when we need to validate some imaging or something that isn't functional, so we don't need it to be awake. But I've never studied specifically the effect.
I would imagine that could answer a lot of that could help to answer a lot of these anesthesia questions that are confounded by the fMRI complexity, where you have more direct interpretability because of your techniques.
Yes, definitely.
Because you could look at the different drug interactions, and like you brought up obviously the difference between the vasoconstrictor and the vasodilators. You could actually map that space of what it does, at least on the vascular side and using your mapping techniques.
True, and we probably already have a lot of that data just because we've been for debugging in our lab, we've been acquiring a lot with anesthesia.
All right. I don't think we have enough time for another question. So that's the end of our lecture. Thank you so much, Dr. Desjardins. Thank you all for joining us today.
Thank you for listening. Thank you for inviting me.